Hello everyone, this is Mrs. Fan. This is a recorded video for week 6, day 1. First, go ahead and take a minute to update the page number in your digital notebook. These are the dates for the odd period. If you are in my period 1, 3, 5, and 7, here are the correct dates. If you are in my even period, please record these dates. If you are in period 4 or period 6, please record the dates right here. Again, the table of contents is on slide number 3 in your digital notebook. With that, this is a reminder that this Friday will be a minimum day, so we will follow our Wednesday schedule where you will have all your period 1 to period 7, but it will be a shorter class like we do on Wednesday. I'm first going to recap the key points we went over last week, and then we'll go over the assignment that you did. And then after that, we will move on to the Explore Activity 2 for microscope samples. The first key point, virus is not alive because it does not have all of the characteristics of living things and they cannot reproduce by itself. Last time we saw a picture of virus. We saw that inside the virus there is a genetic material, either RNA or DNA. And surrounding it we have a layer of protein layer. It's called protein envelope. And on the right we see a picture of coronavirus and we can also see the protein envelope around that. If we take a look at a cross section of the coronavirus, we can see the layer of protein envelope around it, and inside we can also see RNA. We talked about last time that virus is not considered as a living thing because it does not have all of the characteristics of life. One of the characteristics it does not have is the ability to replicate. So virus, they are not able to make more copies of virus by itself. What they do, on the other hand, is that they infect your cells or any cells of an organism that they infect. They insert their genetic material and they use the tools within your cells to make copies of this virus. So again, the first key point that we learned last week is the fact that virus is not alive. The second key point that we learned is cell is the smallest unit of living things. Now, do you remember what tool do we need to see cells? Cells are so small and so tiny, so we cannot see cells with our naked eyes. What tools do you think we need to see cells? That's right, it is microscope. So let's review our classwork from last week and continue. Let's take a look at your assignment below. We'll start with question 1. This part of microscope moves the stage up and down for focusing. That will be the coarse adjustment knob. This part of microscope adjusts the amount of light on the slide. That will be the diaphragm. This part of the microscope is where you view the magnified sample. That will be the eyepiece. This part of microscope moves the stage slightly to sharpen the focus. That will be the fine focus adjustment knob. This part of microscope is where you place the slide on. That will be the stage. Let's take a look at the next picture. So again, as we saw earlier, we will see the eyepiece right here. This is where you look through to see the sample. We have the stage right here. This is where you place the samples. The base, when you're holding the microscope, you want to put one hand below the base and the other hand holding the arm right here. This is the diaphragm that we mentioned earlier, where you can adjust how much light is going through. And we also see the adjustment knob right here. And depending on microscope, it will either be right here on the top or right here on the side. So let's take a look at the next few questions. Number 1, we have the eyepiece. Number 3 would be the coarse and fine focus knot. 4, arm. Number 6 is stage. And number 8 is the base. Let's go back to your notebook. This will be slide number 8 in your digital notebook or page 2, box number 1 in your physical notebook. The question is, how does the microscope help us to determine whether something is living? Go ahead and answer right here. Why do you think the microscope is important? Go ahead and pause the video and write down your answer. Alright, now let's move on to our next activity. Explore activity 2, microscope samples. You see this in slide number 9 in your digital notebook. On the left, it will have the instruction how to sign up for Gizmo. We already signed up for Gizmo last time already. If you have not signed up yet, please take a look at the description below for the link on how to sign up and also the video on how to sign up. On the right, it is the procedures, the step-by-step guide on how this lab works. Now, when you go to the website, sometimes you might see it will show that time is up. It might also show you that you only have 5 minutes to do it. What you need to do at that time is to click on login on the upper right hand corner. When you click on login, put in your username and your password and click login. Again, if you do not know your username or password, please email me and I can let you know what your username and password is. Once you log in, you will have unlimited time to work on the lab. This is the first page that you will see. Go ahead and click on Show All Samples, and you will see all 16 samples that are available. In the example that I did for you, I did microalgae, so let me show you how that works. Click on microalgae, and then click on microscope. Just like we saw in the microscope assignment, there is coarse focus and fine focus. Coarse focus, it will move the stage up and down. Notice right here what happens when I move the coarse focus. Do you see this one? The stage will go down. Take a look again. If I go right, the stage will go up. Now when you are adjusting the coarse focus, you don't want to put it all the way to the left or all the way to the right. Whenever we are adjusting the coarse focus, we want to move it a little bit at a time. So right here, we can see a more clear image. So we don't want to, again, just move it all the way to the left or all the way to the right, but we want to adjust it a little bit at a time until we get a pretty clear image. I'm going to stop right here. And I want to adjust the fine focus. This one right here, it adjusts the image slightly, so I get a sharper focus, a sharper image. So right here, I'm going to adjust it slightly. This is getting blurrier, so I'm going to move to the other direction. So this is a pretty clear image right now. Notice right here, there's magnification. Magnification is how many times am I seeing the image. Right now, I have the image at 80 times. You can also change it to 160 times or even 400 times. Other part that you can adjust is the stage. You can move the slide to the left or to the right. You can also move the slide up or down. Once you get the image you like, click on the camera right here. And again, you're going to click with 2 fingers to right click on your Chromebook. Go to copy image. If you have another way, say you take screenshot on your computer or laptop, you can also do that. You want to go back to your digital notebook, and this is where you are going to place the image. So notice I put the name right here, microalgae 400 times, and then put the image right here. Next, let's click on test for life. We'll do two tests right here. First, we want to test whether your sample is making energy. Second, we want to test whether it's making carbon dioxide for respiration. First, we're going to turn off the light and we are going to click play. The third column right here, this is the part that we want to notice. Now I put the key for you right here. If it is green, then we put yes. If it is light pink, we put no. So notice right here, again, we want to look at the third column. For energy, we see that it's bright green. So we're going to put yes, that this is producing energy. And on the second one, if it is orange, we say yes. If it is hot pink, we say no. So right here on the bottom one, we see that it's orange. That means that yes, it is producing carbon dioxide. So we are going to go back to the slide and we're going to put yes right here. So this is how to complete your example. Microalgae 400 times. The picture, based on the color, is it producing energy? Based on the color right here, is it producing carbon dioxide? So in that way, you're going to be doing sample 1, 2, 3, 4, and 5. Again, just like we mentioned earlier, there are 16 samples altogether. We did microalgae as an example, so you have 15 choices you can choose from when you're doing your samples. You can choose the samples right here, the magnification, put the picture right here, and based on the color of the samples, you can indicate whether they are producing ATP and whether they are producing carbon dioxide for respiration. To sum it up, you are going to finish up to slide 17 today. Again, the earlier slide, we did it in earlier class last week already. So if you finish earlier, you can also double check that you have slide 2 to 17 finished so far. And on the physical template, that will be equivalent to page 1 and page 2. If you're done, again, make sure to check your grade on School Loop. I did update your grade this weekend. Double check, do you have any late work that you need to complete? And of course, you can take a break if you need to during the independent time as well. Have a great day. See you tomorrow.